In this video, we'll talk about Griffith's experiment and how it contributed to our understanding of heredity. How do we inherit traits from our parents? It's a question that long perplexed scientists until the answer was eventually found in deoxyribonucleic acid, aka DNA. We now know that DNA is the hereditary material of all living organisms. But in this video, we're casting our minds back to the early 20th century, a time when even the best educated scientists couldn't identify the mysterious hereditary material. Meet Frederick Griffith, a British bacteriologist. In 1928, Griffith designed an experiment to develop a vaccine for bacterial pneumonia. Instead, he made an alternative but nevertheless valuable discovery. Griffith knew there were two different strains of Streptococcus pneumoniae, the bacteria that causes pneumonia. There's the R strain, which has a rough appearance, and the S strain, which has a smoother outer coat. When Griffith injected a healthy mouse with the R strain, the mouse was fine. It did not catch pneumonia. So, he concluded the R strain is non-virulent. When Griffith injected a healthy mouse with the S strain, the mouse caught pneumonia and died within a few days. The S strain is virulent. Moving to the next stage of his experiment, Griffith took the living, dangerous S strain and killed the bacteria with heat. As expected, when a mouse was injected with the heat-killed bacteria, it lived. The final step of Griffith's experiment was when things took an unexpected turn. Griffith mixed the living R strain bacteria, that's the non-disease-causing strain, with the heat-killed S strain. Even though neither of these strains killed mice on their own, mixed together they did. And when he isolated colonies of bacteria from the dead mice's blood, he saw that they contained living S strain bacteria with their characteristically smooth coating. What did all of this tell Griffith? Clearly, the heat-killed S strain can transform the non-virulent R strain into the smooth-coated virulent type. Griffith named the process transformation. A transforming principle, he said, is transferred to harmless bacteria to make them deadly. The question remained, what could that transforming principle be? What kind of biomolecule or substance changes the characteristics of an organism? A protein? A sugar? RNA? Though Griffith didn't have all the answers and never developed that vaccine for pneumonia, his experiment was the first in a series of landmark experiments. Today, we know the answer. It's DNA. The DNA of the heat-killed strain survived the heating and was taken up by the living R strain, changing its morphology and physiology. But it wasn't until 16 years later that DNA was identified as the transforming principle in another milestone experiment building on Griffith's work. Three scientists, Oswald Avery, Colin McLeod, and Macklin McCarthy, sought to find out if transforming principle was protein, RNA, or DNA.
Griffith experiment. Griffith in the year 1928 was working on a bacterium known as Streptococcus pneumonia. Streptococcus pneumonia has two types of strain, smooth strain and the rough strain. Cells of the smooth strain have a protective layer known as the capsule, which protects the cells against the immune system. This makes the smooth strain highly pathogenic. The rough strain lacks capsule. Hence, the immune system can easily kill the rough strain. When the cells of the smooth strain are injected in mice, the mice is killed. Whereas when the cells of the rough strain are injected in mice, the mice survives. In the next experiment, the cells of the smooth strain were killed by heat. When these cells are injected in mice, the mice survives. However, when the cells of the heat-killed smooth strain are mixed with the rough strain and injected in mice, the mice is killed. And this result was surprising. This means something was getting transferred from heat-killed S strain to live R strain, which converted the live R strain to S strain, which was highly pathogenic. and this phenomenon is known as the transforming principle. Griffith did not know what is getting transferred from the heat-kill S strain to the live R strain. Further experiments to find the transforming principle was done by Avery, McLeod and McCarthy.